Hello students. Again today after because of the Zoom app we were unable to contact with you, but I tried to give my lesson through this recording. Yesterday I gave you the question regarding the chapter 2. We were doing chapter 5, that is structure of cell. Structure of cell. I've told you that the cells are of different shape, size. Some are unicellular, some are multicellular, and even I've told you that what different scientists did work for. They like what Rudolf Virchow did, what Robert Hooke did, what Robert Brown did, who discovered, who invented the microscope, then electron microscope. An electron microscope is a very good microscope through which you can see the different things. One thing more, before starting the chapter, I want to ask you, really you might be feeling very bored as you are staying at home, you are unable to meet with your friends. So this is the leisure time, but apart from studying, there's a leisure time that you can come up with your hobbies. You might be having a lot of hobbies with you. You can learn many things from that. So it is a time that you should groom your hobbies, and really they are very good to spend your time. Some might like writing books, some stories, some poem, like this you can do. So this is the time you can utilize, or if you love gardening, you can do that, or you can help mother, and even you can learn how to wash clothes, because now you are in class 9 and you can help mama in the all day course. So this I want to convey. And what lesson I'm going to give you, that this basically that the cell is mainly made up of 3 parts. And what are the 3 parts it is made up of? One is plasma membrane. Second is cytoplasm. And the third is nucleus. These are the main parts of cells. Now what we are going to discuss today, that is we are going to discuss regarding the plasma membrane. Actually you might have seen this cell. It is very simple, that like this you just see that it is a cheek cell. This is the nucleus present over here, and there's a dots are present over here. So this is cell membrane. We call this as a cell membrane. This is cell membrane. अरे पेन कहाँ गया है इसमें? This is a cell membrane. This is nucleus, and this is cytoplasm. So this is the basic structure of all cells. Now what happens, that in what we are going to read is today, that is plasma membrane. The main topic is plasma membrane. Plasma membrane is also known as selectively permeable membrane, selectively permeable membrane, or this is also known as semi permeable membrane. What does it mean? What this term, ये इसका मतलब क्या है कि सेमी परमेबल, परमेबल या सेलेक्टिव परमेबल? इसका मतलब है कि it allows only the selective things to enter. So what is the function of this plasma membrane? It allows only the selective things to enter. ओके, जैसे अब आपको ज़रूरत है, अगर कोई पार्टिकल का साइज़ बड़ा है तो ये मेम्ब्रेन क्या करता है, आपकी स्किन पे हर चीज़ तो नहीं है कि अंदर है, वैसे इस मेम्ब्रेन से क्या होता है कि वही चीज़ अंदर जाएंगी जो कि इनकी ज़रूरत है, अदरवाइज इट डज नॉट गो इन. द क्वेश्चन इज ऑफन आस्क्ड why plasma membrane is known as the selectively permeable membrane, because it allows only the selective things to enter inside the cell. For this I've bought so many links for you, so you can watch those videos. I'm just showing you.
Plasma membrane and its functions. What is a cell made up of? While studying the cell under a microscope, we will find three common features in almost every cell: plasma membrane, nucleus, and cytoplasm, which are responsible for all activities inside the cell and interactions of the cell with its environment. Plasma membrane. This is the outermost covering of the cell that separates the contents of the cell from its external environment. The plasma membrane allows or permits the entry and exit of some materials in and out of the cell. It also prevents movement of some other materials. The cell membrane, therefore, is called a selectively permeable membrane. Diffusion. Some substances like carbon dioxide or oxygen can move across the cell membrane by a process called diffusion. This is spontaneous movement of a substance from a region of high concentration to a region where its concentration is low. Thus, diffusion plays an important role in gaseous exchange between the cells as well as the cell and its external environment. Plasma membrane and its functions. Osmosis. The movement of water molecules through such a selectively permeable membrane is called osmosis. Thus, osmosis is the passage of water from a region of high water concentration through a semi-permeable membrane to a region of low water concentration. In this process, the solution can be of three types: hypertonic solution, isotonic solution, hypertonic solution. Hypertonic solution. If the medium surrounding the cell has a higher water concentration than the cell, meaning that the outside solution is very dilute, the cell will gain water by osmosis and the cell is likely to swell up. Such a solution is known as a hypertonic solution. A hypertonic solution. If the medium has lower concentration of water than the cell, meaning that it is a very concentrated solution, the cell will lose water by osmosis and the cell is likely to shrink. Such a solution is known as a hypertonic solution. Isotonic solution. If the medium has exactly the same water concentration as the cell, there will be no net movement of water across the cell membrane and the cell stays the same size. Such a solution is known as an isotonic solution. Functions of plasma membrane. Unicellular freshwater organisms and most plant cells tend to gain water through osmosis. Absorption of water by plant roots is also an example of osmosis. Thus, diffusion is important in exchange of gases and water in the life of a cell. The cell also obtains nutrition from its environment. Different molecules move in and out of the cell through a type of transport requiring use of energy in the form of ATP. The plasma membrane is flexible and is made up of organic molecules called lipids and proteins. The flexibility of the cell membrane also enables the cell to engulf in food and other materials from its external environment. Such processes are known as endocytosis. Amoeba takes its food through such processes.
Now read anytime, anywhere with the free Kindle app. Download now.
Brutal sexual assault हुआ है.
One more that I will give you more links of this. You can do videos.
Hello there. I know you wanted to learn about diffusion and osmosis. But before that, you need to know something about concentration. And to understand concentration, we need some salt and water. First, what is concentration? Concentration is the measure of one particular substance in a solution. Not very clear, right? That's why we have the salt and water. Let's take a beaker full of water and add a few spoons of salt to it. Now this here is a solution. There are two substances in this solution. One is the salt and other is the water. We earlier saw that the concentration is the measure of one particular substance that is found in a solution. So the measure of salt present in the solution is the salt concentration, and the measure of the water present in it is the water concentration. As simple as that. Okay, now that you have an idea of what concentration is, let's understand diffusion. We are going to take another beaker, fill it with water. This time we are going to add colored salt. Notice that I am adding it to one end of the beaker. In some time it slowly starts to move, and after a while you can see that it is spread throughout the beaker. This means that the salt, which was initially at the higher concentration at one end of the beaker, slowly started to move to the other end where it was present in lower concentration. This movement of substances from the region of higher concentration to the region of lower concentration is called diffusion. Usually, gaseous exchange across the cell membrane takes place by diffusion. You can also see diffusion happening around you every day, like when the delicious aroma of food spreads from the kitchen. But before you run into the dining room, let's quickly discuss about one more concept, that is osmosis. Again, let's take a beaker and fill it with water. We are going to divide this beaker into two compartments by introducing a semi-permeable membrane. Let's name these compartments A and B. Water can move through the semi-permeable membrane. Therefore, free movement of water molecules occurs across the membrane. As of now, the water level inside both the compartments is the same. Now, let us add few spoons of colored salt to compartment A. The salt cannot move across the semi-permeable membrane and gets dissolved in A. Now that we have added salt only to compartment A, we can say that the salt concentration is high in compartment A and nil in B. Now, if you observe compartment A, out of all the water molecules, some are interacting with the salt molecules and the remaining water molecules are free. Whereas in compartment B, all the water molecules are free as there is no salt added. So, we can say that the free water concentration is higher in compartment B than in A. And we know that compartment A has more salt concentration than B. Now, if I apply the same principle of diffusion, that is, molecules move from regions of higher concentration to lower concentration, how do you think the water will move? Will it be in the direction of A to B or B to A? Obviously, since a higher concentration of free water is in B, it would be from B to A. We use a special term for this type of movement of water. It is called osmosis. We can define osmosis as the movement of water molecules from the region of higher concentration to the region of lower concentration across a semi-permeable membrane. As we know, the cell membrane is semi-permeable. So, it cannot let in this salt X, but it can let in water molecules. How would the transfer take place according to the osmosis principle? Well, there are three possibilities. First possibility is, if the water concentration in the surrounding salt solution is exactly the same as that inside the cell. In such cases, the surrounding salt solution is said to be isotonic. No movement of water will occur in this case. Second possibility is, if the water concentration in the surrounding salt solution is more than that inside the cell. The salt solution in this case is said to be hypotonic. Here, the water rushes into the cell, causing the cell to swell. This movement of water into the cell is called endosmosis. The term endo means entry. The third possibility is that when the water concentration in the surrounding salt solution is less than that inside the cell. Here, the salt solution is said to be hypertonic. Therefore, water moves out of the cell, causing the cell to shrink. This movement of water out of the cell is called exosmosis. Exo means exit. Osmosis plays an essential role in living organisms. For instance, the absorption of water in plants by roots and the absorption of food in freshwater unicellular organisms like the amoeba happens due to osmosis. Now, let's quickly recap what we have learned. Concentration is the exact amount of one particular substance that is found in a solution. The movement of substances from the region of higher concentration to the region of lower concentration is called diffusion. The gaseous exchange between the cell and environment takes place by diffusion. The movement of water molecules from higher water concentration to lower water concentration across a semi-permeable membrane is called osmosis. The cell, when placed in hypotonic solution, swells because of endosmosis. The cell, when placed in hypertonic solution, shrinks because of exosmosis. Osmosis helps the absorption of water in plants and absorption of food in freshwater unicellular organisms. I hope you have learned something new and interesting today. We will meet.
Now one more link I have searched for you. You can go through that, and looking at 1 or 2, 3 times you will learn what is given over here. And really they are very good. One more I have done for you. Yes, right. But more I have wait. I'll just take it off from plasma membrane.
Imagine if you could design products as quickly as your ideas flow. Imagine if 3D modeling was as easy as drawing with a pencil.
In my second year of teaching, I was looking to improve an osmosis lab I had done the year previously. Osmosis, if you remember from our osmosis video, involves water traveling through a semi-permeable membrane, potentially a cell membrane. And I wanted a cool way that students could model this in different scenarios. And one of my colleagues told me about this egg lab. I won't get into the whole lab though. It was actually one of the first steps of the procedure that got to me. To get ready for the lab, my colleague told me, you can soak eggs in vinegar for 24 to 48 hours and the shell comes off. Oh, so I need to make hard-boiled eggs then. No, raw eggs. But if the shell comes off, well, that's the whole point. What's underneath the shell is going to mimic a cell membrane. It's kind of modeling how a cell membrane would function. You know, if the whole egg was actually like a cell. So then you run different scenarios with it for your osmosis lab because it will be a semi-permeable membrane, like a cell membrane. I couldn't visualize this. If the shell comes off, but it's a raw egg, how does it stay together? So I have this area in my house that's designated for things for me to try out. Don't worry, I always clean up afterwards. I tried this experiment out in advance, just to be sure. The hard shell is removed, but the membrane that was always there remains. We often visualize the membrane of a cell this way, like this membrane around the chicken egg. The cell membrane is semi-permeable, meaning it lets some materials through, but not others. We have an entire video all about cell transport and how materials can pass through the membrane. A cell could never be as large as a single chicken egg, though. Why? Well, it turns out surface area is a very important thing. Remember that surface area determines the surface measurements of that cell membrane, and the cell membrane controls what goes in and out of cells. That includes food coming in, as well as molecules that are essential for metabolic processes, and then also waste going out. If volume, which is all this space inside, increases, then you will have more need for surface area, as you will have more need for food to enter, more of a need for waste to be removed, and more metabolic reactions occurring in this larger volume in the first place. If we do a little bit of math here between these two models, I'm going to use popular cube models instead of egg-shaped models because it's a little faster for me to do surface area and volume calculations. See how here there is a big difference in surface area to volume in the smaller model? 6-to-1 ratio. That means the surface area in this small model is 6 times more than the volume. Look at this beautiful ratio with so much surface area. But if we look at this bigger cube and we do some math for this model, that surface area to volume ratio decreases. Sure, the surface area is still larger than the volume in the large model, but it's only 2 times as large now, not 6 times as large. Cells are way smaller than this small model here to allow for an exceptionally large surface area to volume ratio. And a major reason why we're not going to find a cell as big as this chicken egg here. Surface area is important. And while we can model a lot of the processes of cell transport from this egg membrane and how important the membrane is, I don't want to neglect talking about the amazing cell membrane structure itself, because the cell membrane structure truly is magnificent. And since every single living thing is made up of one or more cells, which is part of the cell theory, it's a big deal because every single cell has a membrane. It doesn't matter whether you're talking about bacteria, or protists, or plants, or animals, or fungi. Even archaea aren't too cool to have a membrane. They all have a cell membrane. The structure can vary some, but we're going to be talking about some major structures of the membrane that you can actually find in most cells. We should mention that the fluid mosaic model is often what we use to describe the cell membrane. A mosaic, in case you've ever created one, we did in some of our art classes over time, arranges many small pieces together to make some large piece. You'll see that that makes sense when describing the membrane in a minute. The word fluid implies movement, and this is true for the cell membrane as the components are floating around, they're not static. So let's take a look at some of these components in the cell membrane. We're looking first at a phospholipid bilayer. A phospholipid is a lipid, but an interesting one. So when you talk about a lipid in general, many lipids are nonpolar. Think of oil, for example. It's nonpolar. It won't dissolve in water. Water is polar. But a phospholipid is interesting because one part of it is polar, the head, and the other part of it is nonpolar, the tail. It's amphiphilic. Let's explain what we mean. We often refer to the polar head of the phospholipid as hydrophilic, which means that part loves water. Well, you know, if a lipid could love. The nonpolar tails are hydrophobic. They do not like water. These phospholipids arrange themselves into a phospholipid bilayer with the nonpolar areas here in between, away from any water. It also allows this area in between to be separated from the outside and inside. Water can be found on the inside and outside areas. Also, these phospholipids, they don't just stay put. They move around. It's the fluid mosaic model after all. This gives the cell membrane flexibility. Phospholipids can even flip-flop around, but that's far less common. Remember that this entire phospholipid bilayer borders the whole cell. It would be a sphere even though we're just looking at one area of it. We have an entire video that talks about which molecules can go in and out of this membrane and which ones can't that you can view, but for now, we're going to look at some of the other structures more in depth. Cholesterol. You know, cholesterol often gets a bad reputation. And while cholesterol that builds up in the arteries can be a problem, cholesterol in your cell membrane is critical. If temperatures drop, the cholesterol can actually function kind of like spacers between these phospholipids, keeping them from becoming too packed. Or vice versa, the cholesterol can actually function to connect phospholipids, to keep them from becoming too fluid in warm temperatures. Proteins. In protein synthesis, we talk about why it's so important for cells to make proteins. Many proteins are found in or on the cell membrane, and they play major roles. Peripheral proteins, like the name suggests, tend to be on the peripheral area of the membrane. So while they're on exterior areas of the membrane, they generally are not going to go through the membrane. That's for integral proteins. Integral proteins go through the membrane. Oh, and peripheral proteins can sit on them sometimes. Because of location, these proteins tend to have different functions. Integral proteins, with their potential to go through the membrane, are frequently involved in all kinds of transporting methods for all kinds of materials. Some relevance? Consider the breakfast you ate this morning. Your body digested what you ate for breakfast to obtain glucose. Once in the bloodstream, those glucose molecules can't just squeeze through the phospholipid bilayer to enter all of your cells. The glucose molecules are too big and polar. But your cells need glucose to survive, to make ATP, and they rely on integral proteins to get it. Peripheral proteins tend to be more loosely attached, since they're generally not stuck in the membrane. They have an assortment of functions such as acting as enzymes to speed up reactions or attaching to the cytoskeleton structures to help with cell shape. Both protein types can have carbohydrates bound to them, which can make them considered a glycoprotein. If the carbohydrates attach to a phospholipid, you could have what's called a glycolipid. Glycoproteins and glycolipids can identify the cell as belonging to the organism, self and non-self recognition, which is very important when you're fighting pathogens. They can also be involved in many kinds of cell signaling. In fact, here's some relevance. A glycoprotein known as CD4 is found on the surface of some of your immune cells. The CD4 glycoprotein is essential for some of these immune system cells to interact with each other and activate. However, it's also exploited by the HIV virus. The HIV virus uses that CD4 glycoprotein as a way to bind to helper T cells, which it then can infect. Understanding the components of the cell membrane and how those components are involved in recognition and cell signaling is critical to understanding how to fight back against many viral and bacterial diseases. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.
So this is the videos which I have shown you, and they are very helpful, so you can look at these videos. These are the links, and I have shown you the videos. So if you look at these videos 2 or 3 times, it will make you clear what plasma membrane are, and what is tonicity, what is hypertonic solution, what is isotonic solution, as all things will be clear to you. Now what I want to that after doing this, what you have to do, you have to do the questions given on page, in text questions given on page 61. Questions on page 61, and these are the in-text questions. Apart from this, what you will write, some extra questions I am going to give you. First question is, why plasma membrane are known as selectively permeable membrane. Actually this should be the second question. The first question should be like this: what cell is made up of? Now that the third question is, you have to write regarding this. Define osmosis diffusion and tonicity. Fourth, differentiate between osmosis and diffusion. Fifth question is, what do you understand by exo and endo osmosis? Now sixth question is, explain the structure of plasma membrane. Seventh is, write the functions of plasma membrane. So these are the questions which you have to do regarding plasma membrane. First, I have given you the introduction. You will just watch these videos and you will attempt those questions. Okay, and if there's any difficulty, you can ask me. Is it clear? Now lastly, I want to close by this video by a motivational song. आशाएं आशाएं
Street Dancer 3D. Stream कीजिए सिर्फ Amazon Prime Video पर.
Listen to the latest trending Bollywood songs for free only on Ghana. Download the app now.
Subscribe now and press the bell icon. Never miss an update from Tips Official.
Listen to this.
कुछ पाने की हो आस आस कोई अरमा हो जो खास खास आशाएं हर कोशिश में हो बार बार करे दरियाओं को आर पार आशाएं तूफानों को चीर के मंजिलों को छीन ले आशाएं ले दिल की उम्मीदें हंसे दिल की अब मुश्किल नहीं कुछ भी नहीं कुछ भी आशाएं ले दिल की उम्मीदें हंसे दिल की अब मुश्किल नहीं कुछ भी नहीं कुछ भी
So this is all for today.